Dear colleagues, ladies and gentlemen, let me introduce the results of my research of the topic, soil's biological activity in uneven-aged logged forests in South Taiga, European part of Russia. The relevance of the topic is due to the little and insufficient data on the biological activity of soils and cuttings and their dynamics over time in the southern Taiga of Russia. The purpose of the research is to study biological activity of soils on cuttings of different ages forming single demutation series. This research was done at the Moscow State University Scientific Field Station Satino, located in the Central European part of Russia in South Taiga near the southern border of the Albic Retisols subzone. On the slide, the main characteristics of this area are represented. As you can see, broad-leaved coniferous forest in combination with boreal coniferous forest prevails there, and the soils are functioning under permanent intensive loggings that are already carried out longer than 200 years. The research subject is biological activity of upper horizons of Albic Retisols under old-growth forests and uneven-aged logged forests with spruce plantings. Biological activity of soils is examined according to the following indicators: cellulitic activity, urea decomposition rate, earthworms population rate, and microphototroph species composition. Albic Retisols biological activity under old-growth forest in Satino is characterized by high cellulitic activity rate, average urea decomposition rates, high earthworms population rate, and average microphototroph species diversity. The predominance of shade-tolerant microphototrophic species and their greatest diversity at a depth of 1 centimeter, 15 centimeters. There are dramatical changes in the habitat of soil biota on the cuttings aged less than 5 years, in microrelief, vegetation, and soil properties. Soil biological activity on the cuttings aged less than 5 years is characterized by relatively high cellulitic activity in the topsoils, minimum intensity of urea decomposition, minimum number of earthworms and their concentration in the upper 10 centimeters of soil, reducing the diversity of microphototrophs, replacement of shade-tolerant species by species resistant to a variety of adverse conditions, microphototrophs concentration in the upper 3 centimeters of soils. On the cutting aged 10-20 years, first forest deformation with the grass cover takes place. There is an intensification of the humus formation process. In accordance with this, the indicators of biological activity of soils also change. Soil are characterized by high cellulitic activity in the topsoils, maximum intensity of urea decomposition in the soil horizon A, and low diversity of microphototrophs. Increasing of forest algae species and amphibial species diversity, more even distribution of microphototrophs in the soil profile is observed as well. On the cutting aged 30-40 years, there is a formation of forest type phytocynosis with spruce in the first tier, formation of topsoils typical for forest Albic Retisols. Soil biological activity changes as well. So there is observed minimum cellulitic activity of the soil in the demutation series of cuttings, the intensity of urea decomposition in the soil approaching that in the undisturbed soil, and significant increase in the number of earthworms in the upper part, 0-30 centimeters of the soil, and low diversity of microphototrophs. Forest Albic Retisols microbiota starts to recover. Algal cyanobacterial communities, similar to the forest ones, are forming. distribution of algae and cyanobacteria in soil profile becomes typical for soils under the forests. So, my conclusion of the research. Biological activity in soils on areas of deforestation is most of all determined by the stage of forest recovery process. Albic Retisols microbiota composition typical for forest soils starts to recover on average 30 years after deforestation. By this period, the values of some biological activity indicators in soils have approached the ones typical for undisturbed soils under old growth forests. Oh, thank you for your attention. I am happy to join this Congress.